Hi everyone, I'm Emmanuel, the project leader of Freshwater Biodiversity Surveys. I'm from the Department of Biology of the Ateneo de Manila University and Ateneo Institute of Sustainability. In the Biodiversity Lab, we collect samples from rivers, streams, waterfalls. We collect insects from these habitats in order for us to contribute to achieving target 6.6, which is restoration and protection of water-related ecosystems. So what have we accomplished here in our work for the past 5.5 years? Well, coming inside, we have discovered at least 150 new species of macroinvertebrates, primarily insects, but also shrimps, crabs, among other arthropods. You can see here some of the publications of the things that we have discovered in the more recent years. So the question now is, why do we do what we do? Why do we discover insects? What's the point if they're often neglected? It's important because insects are actually giving silent screams. They do serve as clues on the quality of river systems. If they're present, if particular species are present or particular groups of insects are present in rivers, it means that the river is good, it's clean, it's of great quality. Otherwise, if they're not there, then it's unhealthy, it's dirty. And that will pose not only it will have anything to say about the quality of the rivers, but it will also have an impact on the quality of life being lived by the people living adjacent to the rivers. We have to keep in mind that human civilization was built on river systems. Unlike marine ecosystems, wherein people do not reside, marine ecosystem more or less, rivers are more accepted in the field of biodiversity to be place where people reside. That's where they take their bath, that's where they get their food, that's where they do their laundry, etc. And thus, in as much as human activities impact the quality of rivers, the quality of rivers also will have a thing to say about the quality of life being lived by people adjacent to rivers. That's why we are concerned about the rivers. They do serve as by indicators, giving silent screams about the quality of rivers. And it's for us humans to pick up those clues. It's for us humans, what we do in the Biodiversity Lab, to pick up those clues and tell that this river is of good quality because of the presence of certain species we discovered here in our lab. Welcome to our collections room. In the field of taxonomy, you can't tell if a species is new only after comparing it to the other previously described species. So here we have our collections. Okay, so you can see the 10 specimens, the very first of its kind for the different species that we have discovered. And it's important to note this because researchers abroad would come here to our lab in the Philippines to observe these specimens, especially those which are type specimens, the ones identified with red. After comparing with these specimens and detecting whether what they have is new or not, they can now say if it's new or not. We typically do our sampling in the entire Philippines and even in Southeast Asia. And because of that, we have freezers full of sample from all the years of sampling. And you can see here, there are lots of unstudied materials. We actually invite collaborators, undergraduate, graduate students to join us in the lab, to be the boots on the ground, to join us in investigating all these things which have yet to be discovered.
We identify and discover new species all over the country and use this to come up with sound conservation measures. How do we do this? Taxon sampling. We collect from rivers, identify them using morphological and molecular analysis. We have sampled from a lot of rivers all over the country, covering new islands previously undocumented as shown by the colored figures here. Not only are we able to sample more islands, but also greater altitudes. For morphological examination, we use standard microscopic tools and macrophotography. In the context of the pandemic, we came up with innovative lab-at-home setup. We use molecular biological tools to isolate DNA, amplify the gene, and we use bioinformatics tool to analyze the data. Thank you for listening.